Hello everybody, this is Aloysius Guy in a new tutorial, and today's tutorial is about a molecule that is very essential for living cells, and this molecule is the RNA. So today we're going to cover its structure, its synthesis, and also its function, and we will look at the different types of RNAs as well. So before we start this tutorial, please don't forget to look at the description below so you can go directly to the chapter that you are interested in. So to be able to understand the RNA function and structure, we need first to have a very quick glance at the DNA structure. So what we have here is a cell, and within the cell, we have a nucleus. The nucleus have the genetic material within it, and the genetic material in our cells is forming something called chromosomes. Okay, so these chromosomes are nothing but a DNA that is tightly folded and packed together into these fragments. Okay, and this DNA is made up of a sugar phosphate backbone with nucleobases. So a nucleotide is the sugar phosphate backbone with the nucleobase together called a nucleotide. So we have 4 different nucleotides in our DNA, and they are adenine, guanine, thymine, and cytosine. Also, another thing that we know about the DNA is that it's very important for the production of proteins. So what we see here is that within the human DNA, there's approximately 20,000 up to 25,000 protein coding genes. So, and that means that these genes are responsible of producing all variety of proteins that the human cell has. Okay. However, there are some steps that are missing in here, and these steps are essential to convert the information within the DNA to become ultimately proteins. And one of the main steps is our friend, the RNA, and very specifically, messenger RNA. So after seeing that the RNA is a very fundamental step in converting the DNA information to become proteins, and that's why the RNA is very essential to the cell. So now let's have a look at the RNA structure and compare it to the DNA structure and see how different they are. Okay, so the first thing is, as we showed before, that the DNA is a double helix. Okay, that is made up of sugar phosphate backbone with base pairs of nucleobases C, G, T, and A. We already covered that. However, the difference with the RNA is that the RNA is a single-stranded molecule, okay, and also have some nucleobases within their molecules. So the question is now, do the nucleobases within the RNA different? Are they different from the DNA molecule? So also, sometimes you might find that the word nucleobase is referred to as nitrogenous base. So just be aware of that, because some textbooks or other videos might use this term. So let's have a look. So both molecules have cytosine, and both have guanine. Also, both of them have adenine. However, the difference is in the thymine nucleotide. So the RNA does not have thymine nucleotide, and instead they have uracil. Okay, so uracil is a nitrogenous base that is specific for RNA. So that's the second difference now. So the first difference is that this is a single-stranded molecule and also has uracil instead of thymine. And the third difference is that the naming itself. So we can see the RNA is ribonucleic acid, however the DNA is deoxyribonucleic acid. So why this deoxy is added in the name of the DNA? What does that mean? So let's have a look. Okay, so every nucleotide is made up of 3 different components, okay? The phosphate component, the sugar, and the base, or the nitrogenous base, okay, which is adenine, thymine, guanine, all these bases, okay? So what we see here is that the difference gonna be in the sugar component of the nucleotide. So the ribose sugar would have an extra hydroxyl group. However, the deoxyribose sugar would have a missing hydroxyl group. Okay, so how having this extra hydroxyl group make this ribose sugar different to deoxyribose? So the first thing is that This ribose sugar is found in the RNA. This deoxyribose sugar is found in the DNA. Okay, so how is that affecting the function of the RNA then? So there are some pros and cons of having this extra hydroxyl group. So some of the pros of that is that making the RNA or having much higher reactivity than the DNA. And this increased reactivity makes the RNA able to fold into very complex 3D structures. And these different 3D structures are very important for RNA biological function, especially when it comes to gene regulation. However, there are some drawbacks of having this extra hydroxyl group, is that first of all, it makes the RNA less stable. Okay, which means that it is more susceptible for degradation by ribonucleases. Okay, and also all that would result in having a shorter half-life compared to the DNA. Okay, so now we see how just having this extra hydroxyl group would make the structure and biological function of RNA completely different to the DNA. So, due to the high instability of the RNA molecule, the RNA start forming what is known as secondary structures. Okay, so these secondary structures make the RNA more stable and therefore allows it to stay for longer. Okay, otherwise the RNA will be degraded very quickly. Okay, so there are a variety of different RNA secondary structures, but here we are just showing three of the most common secondary structures for RNA. So the first one is the stem loop or the hairpin structure, and then we have internal loop and the bulge. Okay, so when it comes to the hairpin, the hairpin structure is the most common structure, okay, and it's found in a wide variety and a wide range of RNA molecules, and it's very important for RNA folding and stability and function as well. When it comes to internal loop, we can see that it's found in complex RNA structures, and it is such as the ribozymes and regulatory RNAs. When it comes to the bulge, it's a less common structure of secondary RNAs, less common than these two secondary structures, and it's very important and contributing to the structural diversity and flexibility of the RNA. So if we look here, so this is a single strand, okay, so this is not a like double stranded. So we would have a single strand, and then the single strand of the RNA will bind to unfold on itself, and this forms the base pairing and complementary base pairing between the different nucleotides within that strand, forming this structure. And these structures increase the stability of RNA molecules, preventing early degradation. So after we got introduced to the RNA structure and we now understand how what components form the RNA molecule, now we need to understand and to know where do we get RNAs from. So everything starts from the DNA. Okay, so in eukaryote genomes there are areas of the DNA, they are called genes. Okay, within each gene there is different regions, some regions called exons and some regions called introns. So exons are the regions that are known as protein coding regions, and then introns are the non-coding regions. Okay, so in the process of forming RNA, or in this case the messenger RNA, the process starts with transcription. Okay, so transcription is the process of forming what is known as precursor mRNA, and as we can see here is that the DNA information will be transcribed into an mRNA or a pre-mRNA information, and this information will include both the introns and the exons, so the coding regions and the non-coding regions. Okay, and also we can see two flanking areas here. So there is the 5' UTR and the 3' UTRs. Okay, so what are these regions? So UTR stands for untranslated region. So the word translation, it's related to, we're going to cover it in the next video, and it's related to protein production. Okay, so the whole process is that the DNA information will be transcribed into mRNA, and then the mRNA information will be translated into proteins. Okay, However, these regions are not translated into proteins. However, they play a very important role in pre-mRNA processing. Okay, so what happens after that to get to mature mRNA is that splicing and also more processing, post-processing will occur. So splicing is the process of removing the introns, which are the non-coding regions. So they are removed from the pre-mRNA, and now we have a mature mRNA. Okay, so it has only the coding regions. Okay, and when we say post-processing, which means that after the mRNA is formed, there's going to be some post-processing, as we can see here. Okay, so the first one is the 5' cap. Okay, and this is very important recognition signal for translation machinery. So it's, as we said, this is very important for protein production and translating mRNA into a protein. However, the 3' tail, it's also very important for protecting DNA degradation, and it also plays a role in mRNA exports from the nucleus. Okay, so also I need to mention that all this is now happening within the nucleus. Okay, so after the mRNA is formed, it moves out of the nucleus into the cytoplasm. Okay, so now since the DNA transcription process is the main process of converting the DNA information into RNA information, we need to dive into more details in that process. Okay, and you need to bear in mind that we are talking here about eukaryotic cells. We're not talking about prokaryotes because in prokaryotes the process is different slightly. So we are just focusing on eukaryote cells. Okay, so in our cells, DNA transcription is made up of 3 different stages or phases. Okay, so the first phase is the initiation phase, and within that phase, an enzyme called RNA polymerase. So this enzyme is responsible of synthesizing the mRNA. Okay, so this RNA polymerase will bind to a region of the DNA called the promoter. So promoter region is a region that is found at the beginning of the gene. Okay, so if we imagine that this is a gene, if we said that this is a gene, okay, so the promoter is present upstream of that gene. Okay, and yeah, so it's found at the beginning of the gene, and it's very important for the binding of RNA polymerase. So what happens is that the RNA polymerase will unwind the DNA sequence, okay, and then start moving through the gene. Okay, so all that is the gene that is being transcribed into this RNA. Okay, so as the RNA polymerase moves through that gene, the RNA is being synthesized. Okay, so as we can, a very important thing that you need to bear in mind is that the movement of the RNA polymerase is from the 3' to the 5' Okay, however, the synthesis of the RNA molecule, it's from the 5' to the 3'. Okay, so just to make it easier, so these are nucleotides. Okay, so these nucleotides bind to the RNA polymerase, and then they will just attach it to the synthesized RNA molecule. Okay, so as the RNA polymerase will keep going through the gene until they reach a termination site. So within that termination site, the RNA polymerase will be induced to dissociate from the DNA, and then the RNA will be produced and also dissociate from the RNA polymerase. Okay, this termination site, or sometimes called termination signal, is different in eukaryotes and prokaryotes. So in eukaryotes, it involves having the RNA polymerase recognizing specific sequences, and also it needs or it involves the action of specific termination factors. However, in prokaryotes, it's more simple, and it involves the recognition of a hairpin structure. Okay, so this is the process by which at the end we get a pre-mRNA, which then gonna be spliced, and also adding the 5 G cap and the 3' tail. So now let's understand the different types of RNAs that are found in our cells. So there are multiple types of RNA, so it's not just one type. And the first type is the mRNA or the messenger RNA. So this mRNA out of all types is the only coding RNA. And when we say that is a coding RNA, it means that it's the only RNA that it gets translated to produce proteins. All other types, they are not involved in, they do not get translated to produce proteins, okay? The other two types are the tRNA and the rRNA. So tRNA is the transfer RNA, and it's mainly involved in acting as an adapter between mRNAs and amino acids. And then we have the ribosomal RNA, which is involved in forming ribosomes. And then we have the small interfering RNA, which is involved in silencing gene expression, which means that it inhibits or downregulates the gene expression, okay? And then we have the microRNA, which is also involved in gene regulation. So all these 5 types of RNAs are all found in the cytoplasm. So all these types, once they are transcribed from the DNA, they move out of the nucleus into the cytoplasm, okay? The other type is the other 3 types which are found in the nucleus, and they are the small nucleolar RNA, long non-coding RNA, and small nuclear RNA. And they have different functions. So this one facilitates the modification of other RNAs. This one also involves in regulating gene expression. And for this one, it involves in different processes, including the splicing. Okay, so in the next video, we're gonna dive into and give more details about every type and explore the function of everyone and how they work. But here we are just showing very superficially the function and the types. So as we said, in the next video, we're gonna cover the different types of RNA and their functions and how do they work, okay? And also we're gonna cover some common pathways that are used for removing DNA and removing RNA and RNA degradation. And here are some questions to test your understanding of today's tutorial. I hope you find them easy to answer. And with that, we end our today's tutorial. I hope you find it enjoyable and easy to follow. Please don't forget to like, comment, subscribe, and also hit the notification button. And also write in the comments any suggestions for topics that you want me to make video on. Please also, you can go to the description where you can find the time codes, so you can go very quickly to the chapter that you're interested in. Thank you for watching.